The overall goal of this procedure is to isolate human fetal pancreatic cell clusters. This is accomplished by first cleaning and coarsely chopping the pancreas. Next, the pancreatic tissue is digested with collagenase. Then, the digested aggregates are resuspended and plated in petri dishes. Finally, the cells are plated on HTB9 matrix or incubated as aggregates, and an experiment is performed. Ultimately, RT-PCR, Western blot analysis, or immunofluorescence microscopy are used to show changes in gene or protein expression or localization.
The implications of this technique extend towards the therapy of type 1 diabetes because a number of critical biochemical processes that regulate the transition of human pancreatic precursor cells into mature, glucose-responsive insulin-secreting cells in vitro remains unknown. Generally, individuals new to this method will struggle because of incomplete or overdigestion that leads to poor formation of fetal pancreatic cell clusters. Demonstrating this procedure will be Anna Lopez, a technician from my laboratory.
To begin dissecting the fetal pancreas, aspirate liquid and transfer the pancreas to the 60 mm dish with forceps without HBSS and remove any visible splenic, fat, or connective tissue. Use about half a milliliter of HBSS to rinse the pancreas before using forceps to transfer it to a small sterile crucible and into an ice bucket. Place the crucible in a holder for a 50 milliliter centrifuge tube and, with 2 pairs of scissors, vigorously slice the pancreas for several minutes by holding one